Start here. Yeah, so hi everybody. I'm Isabell Hagemann. This is Flo. I like to start today with our case study about tech transfer and using QbDVision. Yeah, this works. Very good. So I will very briefly give you an overview over the biotech world of Bayer, and then we directly go to the case study and the platform process topic as next. So very quickly, you can see a map. I just want to highlight our biological development sites from Bayer. You can see a few spots in Europe and a few spots in US. These are our main sites where we develop biological processes. We are dealing with a quite diverse set of different modalities, so recombinant proteins, antibodies, and conjugates, but also we are in gene and cell therapy and gene editing reagents. We deliver full integrated biologic process development and clinical manufacturing capability from research, target discovery to launch and marketing. And I want to highlight two pictures here out of a few sites we have, but here you can see our cell culture plants with the 200 and the 2,500 liter scale. They are both located in Wuppertal, and they will be in the focus of our case study today. And this brings me actually to the next slides regarding our case study. So starting with the tech transfer topic, so we've heard for a few moments today, we talked about the technology transfer, so giving only a very brief introduction to it. So what technology transfer means is that to control the transfer of products and processes and knowledges, including its documentation from a sending to a receiving site, which will be the 200 liter scale in our case study to the 2,500 liter scale. Often it's for scalar purposes or to manufacture more material. And of course, we have quite a general principles we have to fulfill. So first of all, there's a comprehensive gap analysis, and this will be the focus of our today's talk. That you assess the risk of changes between the sending and the receiving unit, and often a cross-functional team is involved. We have discussed this today a few minutes and hours ago. So actually our case study today will be the documentation of a gap analysis for process transfer and scale-up of antibody manufacturing processes from the development function, so non-GMP, 200 liter, to a GMP site, 2,500 liter, to produce material for clinical studies. So I've brought this slide to give you an idea of what we have done before we have used QbDVision. So we used to use Excel for performing a risk assessment. So of course you find a section on the left for describing the process on the sending and the receiving side, and on the right side you can see fields for risk scores and justifications for detectability, uncertainty, and so on. And as we all know, Excel is not a digital approach in total, so we're all used to it, but it's not what we want to have in the future. We had large project teams who were involved in this exercise, and we discussed the risks line by line, so it was a quite time-consuming approach, and we did not differentiate between platform-specific changes from the sending to the receiving side and between project-specific changes. And we had no tool to constantly track our changes in the platform. And what's the benefit of it? We will talk about it in a few minutes. First off, before we come to this, I would like to show you how we have implemented QbDVision for tech transfer purposes. So what we have done at very first is we have established or implemented a platform process in QbDVision. So this is meant by the left corner. We defined the process of the sending side, so this 200 liter scale, which, where we have a platform process. We defined the process including standard equipment and materials, and we've done it for the receiving side as well. So including the scale-up, the standard equipment for this side, and so on and so forth. And then we did a gap analysis of these platform process. May sound a bit weird, but I would like to give you an example why it makes sense to us. So here is the UV cell path length. This is a parameter referring to the equipment. It's not a process parameter which may be changed due to or based on the molecule itself, but it depends on the equipment, and we have equipment on the sending side and on the receiving side. So this change will come up in every project until we use other equipment. And so we assess this risk, which is not part of the slide, it would be on the right side, but we assess a general risk for this change. And then we move through all our parameters from both sides and did this risk assessment. We did it in a cross-functional group, so with a small subject matter experts from GMP, development, and QA, and the whole thing, so sending and receiving side, including the gap analysis, is now our default process. And why we have done it, this is our starting point for all upcoming projects. So after kicking off a tech transfer, we copy this whole thing as our platform, as our default tech transfer process. This is done by the development representative, because that's the one who defines the process in the beginning. And then the development representative changes all the project-specific changes, so what really depends on the molecule. This is entered, and for any entry which is changed, we change the assessment status from complete to not started. So it's complete by default, by this default platform process, and every time we change something, we put this assessment status on not started. And why are we doing this? With QbDVision, we can use the function of filtering. So we can filter all our process parameters for not started and have a really short list of all our project-specific changes. And now the representative on the receiving side, in our case it's a GMP colleague, goes through these changes. Then he knows where you have to adapt the process on the receiving side. And then we have adapted our platform process on the sending and on the receiving side. The next step is the gap analysis. And here we can use the same function. We can look for the changes. And now we have a very short list, which we have to discuss in our team, where we have really project-specific changes. It can be prepared by individuals or done in the group, but following this approach, we were able to significantly reduce the number of changes we had to access during a tech transfer. And furthermore, we have the opportunity to get process description out of the system for both processes. And we have, with QbDVision, a tool to track our platform changes, so changes in our platform process. And this will be further explained by Florian.
Thank you very much. How much time do I have left? Can anybody tell me? Maybe. I'm not sure how much detail I can provide here. Is it like 5 minutes? I'm looking for Luke. 5 minutes? All right. Okay. So these are you will see now 2 dimensions on this slide. One is that, and that's one goes from left to right, is the project dimension. When we copy a platform project and make a project out of it, that's what actually Isabell explained already. So in that project, we now can track the changes between the non-GMP process and the GMP process, project specifically. We can also, on top of that, see, and that's sometimes of interest, or many times of interest, we want to track back, or we want to see what is the change to the actual platform process at the time where the project was started. So it's not like we have a clear business case for that. We talked about business case driven or technology driven. I'm not sure what this is. I think it's both, maybe, but you can trace back the changes to the platform as well as the changes within the project, and of course you can trace back the changes throughout the different versions of your manufacturing process. So then the other dimension is the platform dimension, and this is really new, and it's still work in progress in terms of integrating it into our organization within Bayer. So we have to really define a workflow how to do that. But we will have a full track record of our platform process in the future as it evolves. So we have now a tool where we can change. So we have a documented platform process in a digital system, and that's first of all that's the first time we have that. And on top of that, we can trace back all the changes that have been done over the course of over time. So this is now the starting point. So we are now at the very top left of the slide, but in the future, like 10 years from now, we will have, I don't know what platform version we will have, and we will be able to trace back everything up until today. And this is now the last layer, and I think this is just optional. With every project that will be created in the future, let's say 5 years from now, you will also copy or be able to copy all the history, or the whole history from your platform, which is basically not really needed. But I just wanted to put it on the slide just to have it complete. But I think for this one, there's not a real business case yet. Maybe there will be. At least it's possible. Okay, coming to the results and what we actually improved. So Isabel said it already, we reduced, basically by having this platform approach, we were able to reduce the number of records or process items that we actually have to discuss dramatically. We didn't reduce the number of people that were involved in the tech transfer. We just reduced the number of items that had to be discussed, because most of the items, because we have a platform, have been assessed already by our SMEs and don't have to be assessed anymore. And it was actually very easy to convince also our QA department to follow that approach. At the beginning, they were a little bit reluctant, but after telling them the whole story, giving them the bigger picture, they agreed, and now we can do it like this. And by that, we could reduce the number of meetings that were previously needed for upstream from 10 meetings to 2 meetings. And that's why we, of course, could reduce the overall costs. So if you look at the numbers, they're a little bit based on assumptions. So we calculated, for example, for $100 per working hour. And that's how we came up with this with these numbers at the end. But basically, bottom line is we reduced the timelines from I think from the saving was 2 to 6 weeks compared to before, and we could reduce the number of meetings and the time spent doing tech transfer. On top of that, we were able to focus more on what actually matters. So what really changes compared to the platform is that what has to be really discussed. So we were able to spend more focus and time discussing that. And of course, we could create our documents using QbDVision in an automated, semi-automated manner. I think there's still room for improvement here, which is the gap assessment and the process description. But I think another big value was also created because when having or establishing this platform, you can add knowledge in your platform project. So when I go back here, where's the pointer? So this is the part where all your knowledge resides in. And you can add knowledge throughout, like all the time, and you can update everything. And this knowledge is transferred to every project in the weekend in the future. And I think this is a great tool to create and mine knowledge. And that's not, I mean, there are numbers, we save time and we save resources, but actually the numbers in terms of how many dollars we saved or euro, that's not a big chunk compared to what other costs are there in the pharmaceutical industry. But creating and mining this knowledge, I think that's something quite innovative. All right, I think I summed it all up already. So what are the next steps? I already said this platform approach still has to be rolled out in our organization. We have to find or define a way how to manage that. What are the checkpoints and what is the workflow to update and govern this process? What's clear is that we will use QbDVision for that. But we have to define the responsibilities and such, and further work on the automation of the document creation. And then we can just keep mining the knowledge by adding it into the QbDVision platform project and benefit from it now and in the future. All right. I think that's it. Thank you very much.